So it's a pleasure to talk to you today a bit about the preclinical requirements to support the development of pediatric medicines. Oops. And the main topics I would like to cover today are timing aspects of preclinical versus pediatric clinical studies, the determining factors for the need or no need of a juvenile animal studies, and the juvenile animal study design, and also most important aspects on the European pediatric regulation, in case you're not aware yet. Then some EMA experience in the area of juvenile animal studies, and then to conclude with some hands-on information on possible early interactions with regulators. Just one remark, in the interest of time, I will not go into great detail for all of the slides, but I thought it's quite good that you have it as reference material in any case. So before initiation of clinical trials in children, the main guideline ICH-M3 recommends that the results of the repeat dose tox studies, safety pharmacology package, genotox tests are available. Also fertility and PP&D studies could provide quite useful information on developmental toxicities. With regards to carcinogenicity tests, that really depends on what the safety concerns are, and also on the duration of the intended therapeutic therapy, drug therapy, sorry. Then the need on juvenile animal study is case by case, and we will come to that in a minute. And of course, of great importance is also the clinical safety data. Now, before continuing on the juvenile animal studies, I would like to just take a step back and remember that children are not small adults. So at time of birth, several organs are still immature, including those responsible on drug metabolism, like liver and kidney, and other organs that take actually up until adulthood to mature. Now, this may lead to an increased sensitivity as compared to adults, as they may be unique toxicities to developing systems, and also the detoxification mechanisms are still immature at very young age. Now, when is a juvenile animal study needed? Most important aspects here are, of course, the age of the pediatric target population. And the potential and identified target organs of toxicity, and the question whether or not they are still immature in the intended pediatric population. So this drives really a lot of the decision of whether or not there is a need for juvenile animal studies. There are also other considerations that could be quite important to prompt a juvenile animal studies, and those would be to further investigate reversibility of a toxicity, or investigate increased sensitivities and establishment of a safety margin. Other important aspects are, of course, considering whether the intended therapy is acute versus chronic. And also, it's important to consider available data from others in the class. However, there's a bit of caution, because not always all members within one class will behave exactly the same in terms of their safety profile, and I'll have an example later on. Yeah. And there is, of course, the guideline on juvenile animal studies, which I put on the top of the slide. Now, if a juvenile animal study is deemed necessary, then it's important that the results will be as conclusive and interpretable to inform on the potential clinical relevance of the findings as good as possible. And as in adults, the species selection is, of course, of great importance, and this is mainly driven also by considerations on the endpoints that you would intend to look into in that study, and on pharmacokinetic and pharmacodynamic considerations as well in that species. The endpoints selected are based on the concerns, so it's rather a targeted design than a screening design here in most of the cases. And for determining the dose of the definitive juvenile animal study, often a short-term dose range finding studies in juveniles, including toxicokinetic, could be of great help. The aim is here to establish a dose response rather in the low dose range, and high frank toxicity is usually not desirable or needed in these studies. And the low dose in these studies should result in the anticipated clinical exposure. The route of administration and dosing regimen should be similar as good as possible to the human situation. The duration of the dosing period and age of the animals are dependent again on the expected target organs and pediatric target population. And for short-term developing organs, these studies could be confined really to the developmental period of these target organs. And for those organs that have a longer developmental period, a study up to adulthood is probably appropriate. And evaluation of reversibility, or long-term consequences could be also of great importance and should be considered. Now the next slide, this is a general graph, and that might be quite useful when planning the age of the animals and the study duration, as it shows the comparative developmental stages of in the different species as compared to pediatric ages. Now I would just like to briefly touch upon the EU pediatric regulation and its requirements. This pediatric regulation entered into force in 2007, and the aim was really to reduce the off-label use in children, and this should be achieved by making pediatric drug development mandatory for those drugs that could be of significant therapeutic benefit also in children. And small, medium enterprises are not exempt from this pediatric regulation because often you have actually very innovative medicines. It could be of great benefit, and advanced therapy medicinal products and orphan medicines are also not exempt from this pediatric regulation. Now, there is a pediatric committee, the so-called PDCO, and they decide upon so-called pediatric investigation plans, or PIPs. And these are really research and development plans that detail the timing and measures for to obtain a pediatric indication. And this means quality, preclinical, and clinical measures. And the timing of this PIP application should be after completion of the adult phase 1 studies. So keep that in mind. And the measures on quality, preclinical, and clinic that have been agreed by the PDCO are actually binding on the companies. Now, as the expertise of this PDCO is mainly on the clinical side, they have helper groups with special expertise. And on the for non-clinical aspects, this is the so-called non-clinical working group. And their role is to consult this pediatric committee and make recommendations on preclinical requirements to support pediatric clinical development. So mainly, are further juvenile animal studies needed or not, or needed at all? The PIP procedure is about a 120-day procedure with a clock stop in the middle, and the non-clinical working group usually discusses relevant PIPs at least once. Now, the non-clinical working group reviews all available preclinical and also clinical safety data and decides if further non-clinical studies are needed. So it's very important that in these pediatric investigation plans, you well describe the available data and justify the need or no need for a juvenile animal study. Now, some EMA experience. I recently did an analysis of the last 8 years, and that showed that about 26% of PIPs contain at least one juvenile animal study. 71% of these PIPs which contain a juvenile animal study were intended for target population, including children 2 years and younger. The majority of these PIPs contained only one definitive juvenile animal study, And out of these, about 76% were in the rat. And only in 4% of the PIPs, there were only juveniles, also juvenile studies in more than one species. In some instances, the pediatric committee issues so-called full or partial waivers for pediatric developments, and they can also be based on safety grounds. So full waivers, 5.2% of those full waivers, 5.2% were for full waivers, and 9.4% of PIP opinions, these were for partial waivers. And out of these, 17 or 22% respectively were also based mainly or in addition to the results of juvenile animal studies. So these results really matter for the decision making on whether or not a waiver is agreed based on safety grounds in children. To learn more about the usefulness of juvenile animal studies in the area of oncology, some non-clinical working group members conducted an analysis of juvenile animal study reports in 19 anti-cancer drugs. So these were mostly targeted therapies such as tyrosine kinase inhibitors and others. And in 8 cases, new target organ toxicities have been identified. In 7 cases, toxicities were observed earlier and at lower exposure levels. These were from 2 times down to 100 times, actually. And in 11 cases, exposure levels in juvenile animals were higher. Now, the regulatory consequences of these were that in 3 cases, an initial pediatric investigation plan proposal was changed into a waiver. In 2 cases, there were also clinical protocol changes in terms of dose levels, and additional monitoring parameters were included. And in 9 cases, there was a marketing authorization submission already of these packages, and that resulted in the incorporation of juvenile animal study data into the SMPC, and in 2 cases, specific warnings and contraindications were included based on these juvenile animal study results. So the lessons learned from this exercise was that juvenile tox studies revealed serious specific safety issues which may have actually been life-threatening in the youngest pediatric population. Juvenile tox studies revealed unexpected toxicities in several cases, and several of those were not related to the primary pharmacology or observed in adults at all. Now, adverse findings in juvenile animal studies have regulatory consequences, as we saw. So sometimes waivers are agreed instead of PIPs, and clinical protocol amendments might be conducted. Then importance for toxicokinetics was identified. So this could help a lot also to see whether dose adaptations might be needed. And again, when speaking about the clinical, to make these studies as informative and conclusive as possible, and later on decide whether these findings could be of clinical relevance, these TK data is really important. Then one of the outcomes was also that multi-tyrosine kinase inhibitors can have quite different safety profiles. So it's quite dangerous to extrapolate results from one multi-TKI to a similar one. So again, said if you know a class, it might not be the same for all of these members within this one class. Now coming to the last part of this talk, which is interactions with regulators. Now pediatrics matters at various stages during the drug development life cycle, as you can see here in green. And I just wanted to show the different possibilities specifically of early interaction with regulators, as marked here in red on the slide. And again, it shows again that, the timing of the PIP submission, end of phase 1, just to remind again. Now, one possibility of early regulatory interaction are these so-called early pediatric interaction meetings, and they're organized by the pediatrics office at EMA. And the objective here is a discussion on the general development strategy of your pediatric development. And it could, of course, also include questions on the preclinical strategy and the need for a juvenile animal study. Another possibility for an early interaction are these innovation task force briefing meetings. And these meetings are a forum for an early dialogue for innovative therapies and technologies, including advanced therapy, medicinal products, and nanomedicines, and others. And the aim is really to facilitate an informal exchange at an early stage of the information and guidance in the development process. And this could include, of course, also to a main extent, preclinical questions. Another possibility is, of course, the scientific advice. And if it's pediatric related, there will be a close interaction between the scientific advice working party and the PDCO. And it's free of charge if the questions are only related to pediatric development. So keep that in mind. And then another possibility of interaction are these so-called PIP pre-submission meetings. However, here the timing is more confined to when you're close to your PIP submission. But you can, of course, in these meetings also confirm that the information that you have put together to justify the need or no need for a juvenile animal study is sufficient. Now, to conclude, juvenile studies can inform on human pediatric drug development and contribute to safeguard the children in trials. The need for juvenile animal studies need to be evaluated case by case and need to take into consideration all available preclinical information and safety clinical information. Juvenile animal study design need to be tailored based on the safety concern and target organs of toxicity and the intended pediatric target population. And there are several early interaction possibilities with regulators, and scientific advice in specific is free of charge, again, if the question only relate to pediatric questions, including preclinic, of course. So take advantage of this. Thank you.